Greetings and welcome to Life Technologies, an introduction to flow cytometric analysis using molecular probes reagents. If anyone should require any operator assistance during the conference, please press *0 on your telephone keypad. It is now my pleasure to introduce your host, Ms. Jolene Bradford. Thank you, Ms. Bradford. You may begin.
All right. Well, thank you for joining me today in the second part of our flow cytometry reagent webinar. The first webinar was given last November and is available for viewing on our website. My name is Jolene Bradford. I am the Research and Development Associate Director of Flow Cytometry Systems located in Eugene, Oregon, which is the home of molecular probes. Before I start the presentation, I'd like to make a few announcements. First, feel free to submit questions during the presentation. I'll try to answer as many of these as time allows at the end of the presentation. It's likely, however, that I'll answer some of these questions after the webinar has ended. At the end of the Q&A period, we would appreciate you taking a few minutes to complete a survey, which is completely anonymous, to give your feedback on the webinar and give you the opportunity to list out topics for future webinars that are of interest to you. Today's webinar is the second in a series that will continue throughout 2012. We will provide technical webinars on individual topics, introduce various procedures, applications, and reagents, and include basic techniques, tips, and tricks for running experiments in flow cytometry. Coming up next in the February timeframe is a two-part series on the basics of flow cytometry. These two presentations will include an overview of basic flow cytometry principle and system components, as well as a review of fluorescence and gating, compensation, and common tools that are used in flow cytometry. Just like the first webinar, this entire presentation can be downloaded in PDF format. You can always come back and listen to the recording using the same link. The recording will be available 24 hours after the live session. So today's webinar will provide an overview of cell health assays that include viability, vitality, and apoptosis. First, we'll talk about how to identify dead cells. These reagents include the traditional cell impermeant DNA binding dyes and the newer fixable dyes. We'll briefly focus on cell vitality and finally talk about apoptosis, often called programmed cell death. We'll discuss a number of reagents that can be used to investigate individual aspects of apoptosis or used in combinations to provide a more complete analysis of the apoptotic process. So let's talk about cell viability. I'd just like to point out that the icon shown here is from the Cell Imaging mobile app, and in the future, look for a mobile app for flow cytometry. There is a continuum of cell health. At one end, a cell is healthy and it is functioning as it is intended. But generally, a cell just doesn't go from live to dead unless there is some sort of a traumatic event. So there are gradations of health that can be measured to the point of a cell having a compromised cell membrane, and that is considered dead. So why is vitality and viability important? Cells are nonspecifically bind antibodies and look like viable cells. This can potentially lead to erroneous results. Dead cells bind all kinds of markers, and excluding dead cells improves the accuracy of the results. So here's an example of how excluding dead cells from the analysis improves the accuracy of the results. Here, lice human peripheral blood was labeled with CD4 Pacific blue, CD8 Pacific orange, and then labeled with our Cytox orange dead cell stain before acquisition on the Attune acoustic cytometer using 488 excitation for the Cytox orange and 405 excitation for the Pacific blue and Pacific orange. The plot A on the left was gated on lymphocytes using a forward side scatter plot only. The live and dead cells are both included in the gate. And you can see that there is an abnormal co-positive population that is seen. In normal individuals, CD4 and CD8 are mutually exclusive. Plot B on the right was first gated on cytox orange negative cells, which are the live cells, to exclude the dead cells from the analysis. And then a lymphocyte gate was used. And you can see that the abnormal co-positive population is gone. So why use a viability indicator? Here's another example, which is published by the Roeder Lab at NIH, that illustrates the value of using a viability reagent, in this case, one of the fixable dead cell reagents. This group works with frozen leukocyte preparations and used to routinely use scatter gating to exclude dead cells. However, they did some experimentation and treated the cell preparation with a fixable violet dead cell reagent and then stained with their antibody cocktails. And they found that within the a reasonable scatter gate still contained a significant number of dead cells. In the right-hand panel, we can see the results from the population where there is an interest in detecting a very small population. The upper plot shows the results when the dead cells have been excluded, and the lower plot actually is using identified dead cells. And you can see there is a significant difference there. So viability and vitality are principally used to enumerate the proportion of live and dead cells within a population. The diversity of live cells within their environment makes it difficult to devise a single assay that is applicable to all cell types. Viability is often referred to, at least in the flow cytometry world, to the integrity of the plasma membrane. The cell membrane is intact in a healthy cell and acts as a barrier to dye entry. The most common definition of a dead cell in flow cytometry is when the plasma membrane is compromised. When it is compromised, it does allow the dye to label intracellular components of the cell. Cells that successfully exclude these reagents, such as the cytox dyes or propylium iodide, are considered viable. Vitality is often referred to cells that display active metabolic or energetic processes. And there is obviously a gray zone in between these determinations. Some cells may show enzyme activity after the plasma membrane starts to degrade, and some cells may lose some metabolic function before the membrane damage becomes apparent. Impermeant nucleic acid dyes penetrate cells with a compromised cell membrane to exclude nucleic acid, but do not cross the membranes of live cells. So these can be used to identify dead cells within a population. If a cell is fixed, many of these dyes are also used to quantitate DNA content within fixed cells. So there is the traditional propylium iodide, or PI, and 7-AAD. We also offer a cytox series of dyes for easy multiplexing within many experiments. So for the 488 laser, there is cytox advanced, cytox green, and cytox orange. Cytox orange can also be used with the yellow-green laser lines. Cytox blue can be used with the violet, and cytox red can be used with the red laser. Just to distinguish this from cell cycle or DNA content, these dyes should be looked at in a log scale where the positive cells identify the dead cells. This is different from a cell cycle, which would use a linear scale. So here's an example of one way to gate out dead cells from an analysis. This is using peripheral blood mononuclear cells, or PBMCs. And there is a significant dead cell population that is somewhat difficult to determine just by scatter, as you see in the upper left plot. The cells are labeled with a CD4 antibody and with cytox red. The upper right plot shows cytox versus forward scatter to identify dead cells and place a region around the live cells for gating. The lower left plot is then gated on the live cells only, and if you can do a comparison between the upper left and lower left plots, you can see where the dead cells were. And then finally, based on the live lymphocyte gates and the CD4 measurement is made. Cytox Advanced is a dye that has very similar spectral properties to 7-AAD, but it is a much smaller molecule, and so it does reach equilibrium much more quickly. This is an example of using Cytox Advanced along with our Annexin 5 assay, and we'll discuss this a little bit further, but in this case it is not a gating parameter, but it is part of the assay. Our Cytox series of DNA impermeant dead cell stains are available in 5 colors using 3 different lasers. And the Cytox Orange, as I stated earlier, works well with the yellow-green laser lines as well as the 488. And there are choices for the red laser and for the violet laser as well. Another class of dyes for viability detection are the live dead fixable dead cell stains. These can be used to mark cell populations for viability at the time of staining, and then subsequently can be fixed or fixed and permed. These dyes work by a different principle. They are protein or amine reactive. As shown with living cells, the amine reactive dye has access to the surface of live cells and will react only with proteins on the outside of viable cells. However, with cells that have damaged membranes, the amine reactive dye will be allowed inside the cell to react with proteins throughout the volume of the cell. And so the dead cells will be approximately 50 times brighter than the viable cells, and this is seen in the upper right histogram. Since the protein interaction is covalent, staining is not altered by subsequent aldehyde fixation. There are a few things to keep in mind with these reagents. Because they are based on organic fluors rather than DNA binding dyes, they are much easier to compensate out of other channels than the traditional viability dyes. The staining must be done prior to fixation. And because these are live cells, although the live cells are very dim in staining, you have to be aware that the live cells will exhibit some dim fluorescence. Now this does illustrate why fixation is not compatible with the traditional impermeant nucleic acid dyes. Cells were labeled, a mixture of live and dead cells were labeled with either PI or the fixable red cell stain. These are read in the same channel on the cytometer. And you can see in the before fixation that the dead cells are clearly distinguished from the live cells. However, these samples, when they were fixed and then rerun, only the amine reactive dye maintained this distinction between the live and the dead cells. It is lost with PI. The live dead fixable dead cell stains have several advantages. Reduced handling. Formaldehydes are known to reduce risks of viruses. The staining pattern is similar before and after fixation. The cells can be stained and fixed, and then at various times after that, the experiment can be run, including several hours later, without loss of the discrimination pattern. And this is completely compatible with fix and perm procedures. This slide shows a range of fixable viability dyes, and there are selections available for every excitation wavelength and a number of different channels. Here the plots on the left for each color show before fixation and then 18 hours post-fixation to show that the distinction is maintained. As I said, there are 4 laser lines that are very useful here, and some of these reagents are analyzed in traditionally underutilized channels and therefore are of particular value. The fixable violet is read in the Pacific blue channel. Fixable aqua is used in the M-cyan or V500 channel. The fixable near IR is read in the channel used for APC size 7. And if you do have a UV laser, the fixable blue is available for use with UV. So here's an example of the violet fluorescent reactive dye, labeling dead cells before the fix and perm procedure, which is part of the ClickIt EDU direct S-phase measurement. Once again, the identification of dead cells gives us the ability to eliminate them from the analysis. And you can see here the dead cells are colored red. And if you can gate them out, then they are excluded from your analysis. We have tools for compensation for the amine reactive dyes, the ARC amine reactive bead kit. ARC stands for amine reactive compensation. These are optimized for use with each of the dead cell stains and provides an easy method for compensation. We do have sampler kits available for both the live dead fixable dead cell stains and for the cytox stains. It contains one of each color, and this is ideal for use in a core lab. This table shows the range of traditional and fixable dyes that are available. It can help in selecting the dye that would work best in your existing reagents and detector on your cytometer. These dyes are available for pretty much any excitation and laser line, and you should be able to fit something into your existing detectors. And this is a summary slide that you can refer back to. So cell vitality, as measured by intracellular esterase activity, is a recognized parameter of cell health. Live cells are distinguished by the presence of ubiquitous intracellular esterase activity. The acetyl-methyl, or AM, ester derivatives of fluorescent indicators make up one of the most useful group of compounds in the study of live cells. Modifications of the carboxylic acid with AM ester groups results in an uncharged molecule that can penetrate cell membranes. Once inside the cell, the lipophilic groups are cleaved by nonspecific esterases, resulting in a charged form that is retained in the cells. The calcine AM ester is colorless and essentially non-fluorescent until hydrolyzed. The cleaved calcine is well retained within live cells and produces an intense uniform fluorescence. We have 3 calcine AM products. One is available for use with the UV excitation. One is available for use with the blue 488, and the other is with a violet 405 excitation. The plots shown here show a mixture of live and dead cells showing the calcine fluorescence paired with a dead cell stain, either cytox red or fixable aqua. Non-fluorescent rosazurin can be reduced by viable cells to the red fluorescent resirufin and has been extensively used to detect metabolic activity in many different cell types, from bacteria to higher eukaryotes. Rosazurin is non-toxic and stable in culture media, and this allows researchers to continuously monitor proliferating cells and to investigate cytotoxicity in both conventional and high-throughput applications. These plots show rosazurin in combination with cytox green, and you can see a range of cell health is identified. Although we have focused on eukaryotic cells, there are options for testing the cell health of prokaryotic cells as well, as you can see from this listing of products for microbiology applications in flow cytometry. And I just wanted to mention that in the March timeframe, we will be offering a webinar aimed specifically at this topic. So one of the most popular kits is the Live Dead Backlight Viability Kit. And in this case, this uses the Cyto9, which enters all cells, both live and dead, and it's paired with propydium iodide, which enters only dead cells. And you can I just want to point out that this data was collected on the Attune acoustic cytometer, which is pictured on the right here. Most of the data presented in this webinar was generated on the Attune acoustic cytometer. So now we are at our polling question 1, and you can select your answer as soon as you are able to. So the question is, which of the following dead cell dyes are compatible with formaldehyde fixation? And the selections are 1, propylium iodide, 2, 7-AAD, 3, Cytox red, and 4, an amine reactive dye. So go ahead and select your answer. Which of the following dead cell dyes are compatible with formaldehyde fixation? 1, propylium iodide, 2, 7-AAD, 3, Cytox red, or 4, an amine reactive dye. And I'll give about 10 more seconds for you to complete your answer. 5, 4, 3, 2, 1. So I can see from our audience response that 12% of you selected A. Whoops, I'm not seeing the results. Oh, here we go. 18% selected C, 0% selected B, and over 75% of the respondents correctly identified 4 as the response. The live dead fixable or amine reactive dyes do maintain the distinction of live and dead cells. While choices 1 to 3 are all impermeant nucleic acid dyes, these are not as compatible with fixation. All right, so we'll return back to our presentation. And we'll move our focus to apoptosis assays. And the icon here, the RIP, is the icon, again, from the Cell Imaging mobile app. And I'll remind you that a similar mobile app is coming soon for flow cytometry. So the name apoptosis originates from the Greek apo, meaning off or away, and tosis, meaning a falling. This translates to a falling off of petals from flowers or leaves from trees. Apoptosis is distinct from necrosis. Apoptosis was first identified as a distinctive morphological occurrence in the 1960s, and by the 1970s as a well-accepted process. It involves cell shrinkage, cytoskeletal collapse, increased cell permeability, chromatin condensation, DNA fragmentation, cell blebbing, and finally recognition and phagocytosis for removal from the system without inflammation. So here's an example of incomplete apoptosis, where there is a partial separation of digits that has shows the effect of a lack of apoptosis. Apoptosis is normally seen in a number of different processes, such as tissue development and turnover. Defects in apoptosis have been implicated in a number of diseases, either from inhibition of apoptosis or from uncontrolled apoptosis. It has been generally accepted that apoptosis and necrosis are two distinct and mutually exclusive modes of cell death. Apoptosis is frequently referred to as programmed cell death. It is an active and physiological mode of cell death in which the cell itself designs and executes the program of its own demise and subsequent disposal. There are many differences between apoptosis and necrosis. Again, apoptosis is an organized process to remove cells from the organism with minimal disruption of the tissue. This shows an illustration giving a very simplistic overview of apoptosis. Cascade-type pathway pathways are triggered by various insults to the cell. Different triggers proceed through somewhat different combinations and orders of effect, but all involve, at some level, mitochondrial changes, changes in membrane structure, activation of caspases, DNA fragmentation, and ultimately cell death. There are multiple pathways, but they all generally follow a response to an insult that can include changes in membrane structure, mitochondrial structure and function, and ultimately loss of membrane integrity and cell disruption. So I'm going to be talking about a number of different assays. For the most part, I'm going to be using a Jurkat model with camptothecin-induced apoptosis and talking about a number of different assays throughout this process. First, we'll look at the loss of mitochondrial function, often associated in the early stages of apoptosis. Mitochondria are the energy factories of the cell, and this energy production is driven by an electron gradient. Loss of membrane potential is an early and sensitive indicator of cell stress. We will look at detection of membrane potential with the MitoProbe reagents and the MitoTracker reagent. The probes that detect mitochondrial membrane potential are positively charged, causing them to accumulate in the electronegative interior of the mitochondrion. JC1 is a widely used carbocyanin dye in apoptosis studies. It is membrane permeant and carries a positive charge. At lower concentrations, it fluoresces green. At higher concentration, it tends to stack into J aggregates that fluoresce red. If JC1 is used in the correct concentration range, it will stack in active mitochondria and give off red fluorescence. When mitochondria lose membrane potential, the JC1 will leak out, causing it to unstack and cause a shift from red to green fluorescence. With many of the newer instruments, you can also look at a ratio parameter of red to green. The flow plots on the left show Jurkat cells loaded with JC1. The healthy cells in the top plot have a strong red fluorescence, and when the mitochondrial membrane is damaged, as in the lower plot, you can see a shift from the red to the green fluorescence. This change can also be seen in the images on the right. These are 3T3 cells loaded with JC1. The top left image shows the orange mitochondrial fluorescence of healthy cells. And after adding hydrogen peroxide to damage the cells, you can see the pattern change to a diffuse green fluorescence as the JC1 unstacks and leaks into the cytoplasm. A few notes on JC1 usage. This kind of analysis requires careful compensation. JC1 needs to be titered very carefully. Detection of mitochondrial fluorescence is very concentration dependent. JC1 does need to be used at relatively high concentrations. And JC1 does tend to bleed into other channels, so be careful when you multiplex with it. Dye C15 is another cyanin dye that can be easier to use than with JC1. This dye also has a positive charge that causes it to concentrate in active mitochondria. It is red fluorescent and excited with the 633 laser. There is no excitation by the 488, so this dye can be easily multiplexed with other reagents. The overlay histogram shows Jurkat cells stained with the dye C15 before and after treatment with CCCP, and you can see that there is a nearly a decade decrease in fluorescence intensity. And on the right, we can see MRC5 cells labeled, giving a characteristic mitochondrial staining pattern. Another valuable reagent is the MitoTracker red dye. Again, it is mitochondrial selective. It is excited by the 488 laser, read in the channel used for PE. It is also easy to multiplex with other reagents and is shown here with Nexin 5 APC. The left plot shows the MitoTracker with normal cells, and then with CCC treatment, you can see a decrease of fluorescence of the mitotracker red. Here's another example of apoptotic cells with this time using Alexaflor 488 and XN5 along with the mitotracker red. And you can see with the treated cells, there is a decrease of fluorescence in the mitotracker red. Now we'll look at a different mitochondrial event thought to occur early in apoptosis, and this is the permanent opening of the mitochondrial permeability transition pore, or MPTP. Permeability transition pore is made up of mitochondrial membrane components, and it is thought to flicker open to dump excess calcium from the mitochondrial matrix into the cytosol. The function is inhibited by cyclosporin A. The pore has been observed to open permanently during apoptosis. The MitoProbe transition pore assay kit uses a published imaging assay to look at this event by flow cytometry, and it is based on the selective passage of cobalt by the pore. So this illustrates the principles of the assay. Cells are loaded with calcine AM, which passively diffuses into the cytosolic compartments, including the mitochondria, where it is cleaved by esterases to form the fluorescent dye calcine. The fluorescence from the cytosolic calcine is quenched by the addition of cobalt. However, cobalt does not cross into the mitochondria, and the calcine within the mitochondria continue to fluoresce. When the pore is opened with a calcium ionophore, the cobalt then enters the mitochondria and quenches the remaining calcine. And the flow cytometry results can be seen in the histograms at the bottom. Total calcine fluorescence is shown at the left. The center histogram shows the remaining mitochondrial fluorescence after adding cobalt. And the fluorescence has been quenched by cobalt with the addition of an ionophore in the plot on the right, indicating the opening of the transition pore. This next slide shows this assay in an imaging application where BPAC cells are labeled with calcine AM, mitotracker red, and Hertz 33342. And you can see in panel A, there is a uniform fluorescence. When the cytosolic fluorescence is quenched with cobalt in panel B, you see the characteristic mitochondrial staining pattern. In panel C, after adding the ionomycin to open the transition pore, the green fluorescence is gone. The remaining orange fluorescence is from mitotracker red staining. And finally, with the addition of cyclosporin A, this blocks the effect of ionomycin. Loss of membrane asymmetry is characterized by an increased expression of phosphatidylserine on the outer leaflet of the plasma membrane and is a hallmark of apoptosis. We have a number of different reagents to look at changes in asymmetry and increased permeability of the membrane. So in XN5, in normal viable cells, phosphatidylserine, or PS, is located on the inner leaflet of the cell membrane. However, in apoptotic cells, it translocates from the inner leaflet to the outer leaflet of the plasma membrane, thus exposing phosphatidylserine to the external cellular environment. An XN5 is a calcium-dependent phospholipid-binding protein. It has a high affinity for phosphatidylserine. An XN5 can be labeled with a number of different fluorophores to identify apoptosis based on changes in membrane asymmetry. This is illustrated here, where we have phosphatidylserine in healthy cells located on the inner leaflet. And as cells undergo apoptosis, it translocates to the outer leaflet of the plasma membrane and is exposed to the extracellular environment and can thus be labeled with an XN5. And this is fine for cells that have intact membranes. Once a membrane integrity is lost, an XN5 can get into the cell and stain phosphatidylserine on the inner membrane leaflet. So we do want to combine this with a traditional viability dye to resolve cells that have lost membrane integrity as well. So here's an example of annexin 5 versus PI, and you can clearly see that there are three populations are identified: viable cells, apoptotic cells, and dead cells. The untreated cells are on the top, and the induced cells are on the lower spot. This shows another example. It's just using some different colors. In this case, annexin 5 is conjugated to Pacific blue and is used in combination with a Cytox advanced dead cell stain. Control cells are represented in the left plot, and treated cells are represented in the plot on the right. Some tips on using annexin. Calcium and magnesium are required for annexin binding. This binding is reversible, so it does need to be present throughout the entire assay. Analysis should be carried out quickly, usually within an hour, and the sample should be kept on ice until acquisition. Some cells, such as megakaryocytes and platelets, some myeloid lineage cells may have large amounts of phosphatidylserine on their surface. That's just something to be aware of. And again, in advanced apoptotic or necrotic cells, annexin 5 can label the inner membrane leaflet as well. Adherent cells, which are removed by mechanical scraping or trypsin, may flip their residues independent of apoptosis, so careful removal is required for this assay. There are some situations where staining with annexin is not the optimal method for detecting apoptosis. This would include assays where cells are sensitive to high calcium concentrations required for annexin 5, or assays where detection on adherent cells is adversely affected by trypsinization, or where washing of cells is prohibitive. These monomeric cyanin dyes, this would include POPRO1, YoPro1, and ToPro3. They have been shown to penetrate apoptotic cells because of permeability changes of the plasma membrane. These cells enter apoptotic cells and then bind to nucleic acids, while at the same time the traditional cell impermeate stains are excluded. These have unique excitation wavelengths and provide an enhanced flexibility when you're doing multiplexed assays. We have a newer dye, the Violet Ratiometric Membrane Asymmetry Probe. The dye itself is called F2N12F, and you can see the full chemical name there. As stated, normal cells exhibit an asymmetry in the lipid distribution, and when phosphatidylserine and phosphatidylethylamine, which are normally located on the inner leaflet, during apoptosis they will translocate. The Ratiometric Membrane Asymmetry Probe is a novel violet excitable dye that can be used for detection of changes in the membrane asymmetry by detecting variations in the surface charge, and this actually gives two separate emission bands. Now this is a ratiometric dye, and if you haven't had the opportunity to use ratiometric dyes, they're really great. Ratiometric probe is somewhat self-calibrating. It is somewhat independent of probe concentration, dye concentration, cell size, and even instrument variation. And this particular dye only requires 5 minutes of labeling time and does not require any wash. No special buffers are required and can be used with adherent cells very easily. Here we see the orange and green fluorescence in the dual parameter plots on the control and treated sides. The plots on the right use a ratio parameter on the x-axis, and the ratio is shown in a dual parameter staining with a dead cell stain. Again, this data was collected on the Attune cytometer, which does support this derived parameter. The live cells on the lower right quadrant are seen in the lower right quadrant. The apoptotic cells are in the lower left quadrant, and the dead cells are seen in the upper left quadrant. So So this brings us to our next polling question, true or false? Both annexin and the violet ratiometric dye F2N12S are indicators of changes in membrane asymmetry. True or false? Both annexin and the violet ratiometric dye F2N12S are indicators of changes in membrane asymmetry. True or false? And we'll have 10 more seconds for you to submit your answer. 5, 4, 3, 2, 1. And I see that we have almost 80% of our audience selecting true, and that is the correct answer here. Both annexin and the ratiometric dye measure changes seen in membrane asymmetry. And now we'll talk a bit about caspases. These are key components of the apoptotic machinery. We have caspase activity kits to detect caspase 3,7, 8, and a polycaspase. Cell event caspase 3,7 green detection reagent is a novel fluorogenic substrate for activated caspase 3 and 7. This reagent consists of a 4-amino acid peptide conjugated to nucleic acid binding dye. This cell permeant substrate is intrinsically non-fluorescent. After activation of caspase 3 or caspase 7, the peptide is cleaved, enabling the dye to bind to DNA and produce a bright fluorescent signal. There are several benefits of the cell event detection reagent compared to other methods for detecting caspase 3,7. One important advantage of this assay is that no wash steps are required, and this helps preserve the fragile apoptotic cells typically lost in wash steps. There is also the FLICA methodology for detection of caspases through a fluorescence inhibitor. This cell reagent is permeant and contains a caspase recognition peptide bound by a fluorescein and a thiol-reactive fluoromethyl ketone. If a reagent in the cell encounters the appropriate activated caspase, it binds to the active site of the caspase and covalently reacts with a nearby cysteine, locking the fluorescence into the cell, and unbound reagent can be washed out. So this is a plot that is typically seen with a FLICA reagent. When your cat cells are induced with camptothecin, you can see that there is a change from live cells to apoptotic cells. And again, it's a good idea to bring out the necrotic or dead cells using some sort of dead cell stain. Finally, we're going to look at several reagents used to detect nuclear changes associated with apoptosis. These include chromatin condensation and DNA fragmentation. Cells undergoing apoptosis display an increase in chromatin condensation. Morphologically, the nuclei of apoptotic cells become smaller than those of normal cells and become hyperfluorescent when labeled with some nuclear stains. Hertz has long been used to detect this change of increased fluorescence in condensed chromatin, and you can see that in the plots on the left. Hertz is not excited with violet lasers very well, and you can get a better resolution with the dicycle violet dye in the same manner, and these plots are shown on the right. We can see a different effect using the dicycle orange cell permeant dye. Here we can see a sub-G1 population that shows up in late apoptotic cells. In a classic assay, cells are fixed and permed, treated with RNase, and then stained with PI. However, we have stained intact cells here with the vibrant dicycle orange stain and then used a traditional viability dye to pull out necrotic cells. The right-hand histograms showed your cat cells which have been treated with camptothecin for 6 hours and then labeled with the dicycle stain and with Cytox blue as a viability dye. I'm not going to say much about tunnel. These assays require the cells to be fixed and permed and uses a terminal deoxynucleotide transferase, or TDT, to stack BRDU on fragmented ends of cells. And anti-BRDU is used to detect the cells with fragmented DNA dye, while a nucleic acid dye such as PI is then used as a total cell stain. When studying apoptosis, it's very useful to set up a time course to understand when the events in your model occur. And this also allows the cells to be harvested at the same time you run the same experiment on your cytometer. So whenever you are developing an assay, it's very important to understand when in the particular model you need to be looking at the events that you're interested in. So this just illustrates that doing a reverse time course allows you to harvest at the same time and then put them into the same experiments. Nuclear changes. So we see here the appearance of the sub-G1 peak. This is using a couple of different assays at the same time. And this does illustrate a time course from 1 hour to 6 hours of induction. On the lower plot, we see the dicycle violet chromatin condensation assay paired with 7-AAD, and you can see the appearance of the condensed chromatin in green. The top set of plots uses dicycle orange paired with Cytox red, looking for the appearance of that sub-G1 peak. So looking at this time frame of camptothecin-induced Cat model, we see before annexin binding that di-IC15, Mitotracker red, and are able to detect Vim annexin 5 populations pretty much concurrent with annexin 5 binding is the monomeric cyanin dyes and the violet ratiometric dye. We notice that JC1 and mitochondrial assays decrease as annexin increases. Caspase activity tends to increase after annexin, and C12 rosagirin activity decreases after annexin positivity. And of course, the later events of membrane integrity come last. So I'd like to invite you to join our Facebook pages on flow cytometry and immunology. You can find more technical resources by visiting our flow cytometry page there, and it's a great resource to ask some of the questions that you do have about flow cytometry and get some immediate responses. And for your reference, I have products listed here that were used to generate the data in this presentation. This shows cell-permeant nucleic acid dyes, live dead fixable stains, compensation tools, the monomeric cyanin dyes. The next slide goes on to show mitochondrial kits and the membrane asymmetry kits. The annexin list is somewhat incomplete. We do have a number of different conjugates that if you visit our website, you'll be able to see all of the different colors that are available. And then finally, Viability kits, our caspase kits, and some Lage state apoptotic kits. So when you look at the PDF, you can see the listing of these reagents. And that does bring us to the end of this webinar. And I would like to remind you to submit your questions. I thank you for your attention, and I'd like to remind you about our next webinars coming up in February and March. So let me look at some of the questions here. With annexin 5 staining, there is a question about the ability to do intracellular staining. Annexin actually can be fixed. You would have to optimize your experiment very carefully if you were wanting to do that. It does tend to, you have to be very careful in your washing steps. and maintain a high calcium concentration for that. I have a question about what is the biggest advantage of using a cytox dye over PI when gating out dead cells. The biggest advantage is that propydium iodide has a very broad emission, and it does have significant bleed through into the surrounding channels. The cytox dyes offer you different options for moving that to different lasers or farther out in the emission spectra with the 488 laser, and it makes for easier compensation. So I have a question here. What do you suggest to compensate the annexin dyes if you are trying to detect a small population of apoptotic cells? That's a really great question. What I tend to do is have some dead cells or fixed cells that are available, and I label them with the annexin so that it will give me a very bright signal, because if you don't have a lot of apoptotic cells in your normal population, it will be very difficult to detect a positive population. There's a question, can F2N 12S be fixed? And unfortunately, that answer is no, it cannot be fixed. I have a question about why do we not have the same percentage of dead cells with 7-AAD and the live dead kit. I'm not sure which example that was referring to specifically, but they should be very close. Yes, we did focus a lot on mammalian cells, and some of these dyes are compatible with yeast, and I would invite you to come back to our microbiology webinar, and we'll talk more fully about that. And I think that does bring us to the end here. I would like to ask you to stay on the line and complete our brief survey, and thank you for your attention.